So our next speaker is Dr. Tim Hickling of Roche in Basel, Switzerland. Tim, I've given you permission. You can go ahead and start sharing your slides whenever you're ready. As of this month, Dr. Hickling is the head of immunosafety at Roche. He was, of course, formerly at Pfizer, where he was investigating the immunogenicity of biotherapeutics. He was there from 2007 and has worked on early stage vaccines and from early discovery to late stage development of biotherapeutics. He obtained his biochemistry degree in 1995 and immunology doctorate in 1998 from the University of Oxford. He carried out postdoc training at Glaxo and the UK MRC's National Institute for Medical Research before taking up a lectureship at the University of Nottingham, UK. And Tim has worked on various aspects of innate and adaptive immunology in infectious and inflammatory diseases. And he will be sharing his work on the application of immunogenicity prediction and mitigation for development of biotherapeutics. All right, go ahead, Tim.
Hey, great, thank you. So yeah, nicely sort of following on from the last talk, because clearly examples of biotherapeutics where immunogenicity is an issue, clearly in terms of big selling, but big impact therapeutics where we've got many thousands of people on these drugs, and then everything's going along nicely, and then you get an immune response, and unfortunately for a lot of those people, they're no longer able to use the drug and may suffer from some adverse events along the way. So what I want to take you through today is not so much the de-immunization strategy, but using some of the same kind of thinking as to how you actually start the development of a therapeutic with an end goal in mind of having a low immunogenicity. And to highlight here, these slides were prepared my time between Pfizer and Roche, and probably represent more of a Pfizer view of the world. And I'm certainly learning some interesting things about how different companies operate. So, I mean, ultimately, the theory here is, well, everything's kind of better if you design it best in the first place. And certainly those elements of, how do you get enough humanization in a monoclonal antibody with the same things we've just heard talk about, right? Retaining potency, reducing T cell epitopes, and then also maybe a bit more difficult to predict, but reducing B cell epitopes as well. This is actually from a publication from a few years ago where the protein engineering team really led a good effort as to how to go from, on the left-hand side here, some parental clones where you'd got either mouse or rat monoclonal antibodies. They were grafted onto human frameworks, and you can kind of see the sort of boring grey bits are those that the immune system should find boring and grey and not recognise because they're human, whereas the colourful bits are the non-human sequence. So as we go from left to right, grafting on and then applying what's called an augmented binary substitution method, where you literally walk across those CDRs and trying to turn the sequence back into human, you can see essentially the amount of foreign sequence is reduced. So at least one would hope from an ADA perspective that there's less here to see. So that should offer some advantage in terms of reducing B cell epitopes. Interestingly, amongst these particular examples, that didn't always reduce the T cell epitope content. So again, as with most complex development, if you only have your eye on the one problem you're trying to fix, you might miss something else and cause a bigger problem. So in this case here, we had sort of the what's called the parental, and this is in silico through Epivax's ISPRI program, and kind of highlighted some of the changes that can occur, so we can lose some of those sequences that may be immunogenic, but sort of potentially gain some others. So, kind of coordinating how we can get the immunogenicity assays alongside protein design is really key. So that's what I'll kind of focus on for today. There's probably a number of publications and positions on understanding the components involved in immunogenicity, and I'm always conscious of a meeting like this where we've got immunologists with a diverse array of backgrounds. This obviously is an oversimplification of how the immune system operates, but at least in thinking about the tools we have and can look at preclinically, so in vitro assays, we kind of consider our therapeutic protein interacting with our antigen presenting cells, mostly in DC-based assays, and then what actually gets presented to the T cells. I'm sure Jeremy will run through a few of the assays as well in the following talk. Of course, we understand that the therapeutic protein would also be recognized by a B cell, processed in a similar manner, and then where you get the presentation to the T cell, this will obviously help drive things like the affinity maturation and ultimately production of lots of antibodies that will go on and be our anti-drug antibody set. So historically, we kind of started off with, we put protein in and we measure the ADA. And kind of only once we got good at measuring ADA did that really get us to a point where we understood that this is just pretty normal. It's normal biology a lot of the time is what's happening to cause these ADA to appear. And then we can use this to help us understand that process of deimmunization or being better by design to come up with low immunogenicity molecules. I'll somewhat gloss over in silico at this stage. That's not to say it's not important, but sort of concentrate on some of the in vitro assays here. And just to kind of highlight again that probably the history of use of a lot of these assays in recent years has been a lot around molecule selection. And so, often we've ended up on projects where there may be 3 or 4 leads and we're looking for a candidate, and the leads all look remarkably similar. So, it may be that in cases where there is an epitope, well, everything's got that epitope. So, do you really have much to play with? So the opportunity we saw was to really put the in silico in the hands of the protein engineers and say, well, whilst you're just like designing everything else around optimizing your molecule, well, just include this immunogenicity risk assessment. And, sure, it's not perfect. It probably is over predictive. But if we kind of marry this up with a PBMC assay, this then allows us to pick out things that we can see for sure are potential liabilities. So it's always good as we're going through along the project timeline that if there's data we can make a decision on, that can add a lot of value and at least make us feel like we're doing the right thing as we try and aspirationally aim for that 0 epitopes within our proteins. So we got a variant of the MAPS assay, which we've sort of labeled DCMS here. And again, really, if you kind of got to a point where there's still some liabilities that you observe by a PBMC assay, we can kind of load up the DCs with the same protein and see if those peptides are actually presented. And clearly where you've got sort of a PBMC signal or a T cell proliferation signal due to that peptide and the peptide's presented, that's a risk that you want to understand a bit more deeply. So things like a DCT or DCCD4 assay we've found helpful in then trying to discriminate between those leads to go ahead with selecting a molecule for the clinic. Any stage along here, potentially you can select low-risk molecules if you're seeing signals that you're happy with. And also this has to be done in the context of, a whole project-focused immunogenicity risk and mitigation plan. As Amy went through the list of potential issues or potential factors that can contribute to immunogenicity, you really do want to dive down into knowing that you're clean in this sort of more complex system where you've not been able to mitigate against some of those. So just to dive a bit into the assays and say this one here, we actually used our ProImmune's ProMap T-cell proliferation assay. So as I say, we did screen out risk, or we looked to screen out risk by the in silico, and then we take the peptides that we've either not screened out or ones we want to check whether they are actually nice and clean, put those into the PBMC assay and look for T-cell proliferation. You can kind of see on the right-hand side, there's a couple of examples. One where we had a parental sequence that was, remarkably antigenic in the assay, possibly one of the strongest responses we've had in this assay. And then by in silico design, having gone through and selected a few potential alternatives and demonstrating this range of different outcomes in that PBMC assay. So this was actually all the sequences were kind of done on the fly in a project team meeting. Protein engineer, computational protein engineer was also looking at the likelihood of this antibody still binding to its target. And so with the, I think it was like a 10-week turnaround from ProImmune, we were able to get this data back about the same time we'd got data back on potency assays and some of the other assays the protein engineering group were doing. So this kind of helped us to say, well, yeah, look, these are good options, and they were also good options as far as the project was concerned. On a slightly different note, and probably almost always what we'd like to see, is one project that followed both the ABS approach and sort of some kind of intensity on the in silico. We were able to pick out all of these, both framework regions and CDR regions, and putting those into the ProMap assay. We got nothing more than, I think one of the framework here had 2 donor responses, so 2 out of 40 was a 5%. And so we kind of highlighted all this as low risk. And with this particular molecule, we didn't do any further studies. Other aspects of the risk assessment suggested it was low risk. I understand clinical data is about to emerge. I don't want to sort of preempt that too much, but hopefully my former colleagues at Pfizer at some point will return to tell the story of that one. So ProMap helping us identify a low immunogenicity clinical molecule, I think is a good advert. So further on, and say that one of the assays that we like to use as well is the MAPS type assay. And in this particular example here, you can kind of see some of these regions that come up, particularly this heavy 3 region, like in the adalimumab example as well. We typically don't worry too much about the germline sequences. Kind of conscious that some germlines may be rare, and therefore you're not necessarily going to have tolerance in the population. But as Amy was highlighting, some of these sequences are the ones that are flagged as potentially T regitope or Treg stimulating sequences. So we don't rush to kind of change these, but we kind of look at alternatives, obviously, where we've got some sequences. And clearly when you're making changes in the antibody, sometimes for potency, you need to compensate in other places. So some changes here needed some compensation. Interestingly, across these variants, it wasn't just the single change in amino acid that led to presentation or not. In fact, V5 also contained a half-life extending portion as well, which seems to have led to essentially escape from the DCs in the context of this assay, and therefore nothing was presented from this region. So there's quite a lot of things to think about beyond just the sequence, and putting that into practice, I think, is certainly can have its challenges as you're trying to work at the pace of the project teams. So we kind of like looking at the DC CD4. This can get a bit more complex with regard to aspects of biology. Just to highlight with proliferation here, we actually looked at this with an in-house assay where we'd kind of gone with 20 donors, and we're able to highlight some differences. I'll tell you a bit about the story of BOCOCIZUMAB, which was an anti-PCSK9 antibody. An anti-TL1A, so this is in anti-inflammatory, and then Enbrel, which is the TNF receptor coupled to an FC. So these are kind of useful benchmarks that kind of come up fairly repeatedly in this sort of space. And we've got a couple with higher clinical immunogenicity than we like, and an example that's low. I realized that different companies or groups will have their preferred controls in these kind of assays. And sometimes the ones that would look like good controls don't necessarily behave that well in them. So I wanted to go now on to the impact of immunogenicity in this particular clinical example. And this was published in 2017 and is the result from a couple of arms of a phase 3 study. So this is anti-PCSK9. So this is actually the PD marker. We're looking to lower LDL cholesterol. So if I orientate you here, we've got the cholesterol here on the y-axis. We've got the length of study in weeks. So this is a 1-year study with a little bit of follow-up. And then, so the lines relate to the LDL cholesterol as split out by ADA titers. So we've got high titer, medium titer, and then low titer or no ADA. And then at the bottom, we've got sort of the prevalence and incidence of the ADA. So the couple of things I wanted to highlight here were ultimately it was the higher titers of ADA, and this was consistent with neutralizing antibodies as well, but essentially it's the higher titers of ADA where we saw this kind of loss in efficacy. And you can see obviously with low ADA that seems to have no impact over being ADA negative. So this kind of is a good illustration of how we often start saying, well, ADA could have an effect, they might not have an effect. And in this case as well, I think there was a few, do I need to admit this person as well? Sorry. There was a few things around adverse events related to injection site reactions, which were more common in the ADA positive subjects, but again, it's not black and white. So linking immunogenicity to immunosafety are not always go hand in hand that way, as you might expect. But the other piece here then is that you can see that it can kind of take a little bit of time for either the observations of the ADA, so that the prevalence or the percentage any given time point is this sort of lighter of the bars, and the darker bars, the overall incidence. So it kind of peaks in mid-study, and then by the end of the study, nearly half the subjects had been ADA positive at some point. And one of the things to really highlight as a drug development issue is that we can kind of see here that over a 12-week period, although the ADA incidence comes up, the impact on the efficacy is fairly minimal. The paper goes into detail as well about things like drug exposure, and at least it looks to me like it's pretty much in line with you're not seeing so much effect early on. And this can kind of lull project teams into that false sense of security early on. They go, well, we're seeing ADA, but they're not doing anything. Should we worry? My answer is usually, well, it might depend on a range of other factors, but certainly don't get complacent about seeing no impact early on. So in the spirit of kind of looking at what we were doing for BOCOCIZUMAB from a retrospective, and I think this talks a bit again to the de-immunization question. You know, we can kind of look back and go, well, if you look at in silico prediction, if you look at running peptide assays, that there are quite a few potential liabilities. And just to kind of highlight some of the sequences we looked at, there was one particular one in the CDRL2 that gave a fairly strong response. And I think these X's are essentially lined up. So you can see as we walked across it that sort of disappeared. So, you know, we'd highlighted here that there was certainly a liability. At the time, we weren't actually able to find that sequence with a MAPS analysis presented, but you know, we've kind of been doing further work on that, and again, that may be subject to disclosure at a later date. Just to highlight as well, again, you know, there's obviously other risk factors involved here. BOCOCIZUMAB turns out is a little bit on the sticky side, which kind of contributed to things like poor PK. But we also noticed in a DC activation assay that we were seeing, you know, quite a lot of donors sort of in the 30–40% region where you get direct DC activation, and you can see there that potentially you could lower threshold for immune response even if you weren't getting a lot of peptides presented in the context of the MAPS assay. We were able to see in a T-cell proliferation assay as well, and we were looking at some different cutoffs. I know different companies and groups use different cutoffs for this. This was following a robust statistical assessment of the assay. So instead of kind of going for the default stimulation index of 2, we were able to look at, well, actually I've got both 1.5 and 1.4 on here, but we're comfortable that 1.4 is a real result in this assay. So again, it doesn't give us a 50% level, but it gives more of an indication from this kind of data that maybe where bococizumab was heading. And I'm conscious that we're not sort of totally cherry picking from here because we had some similar data with some other immunogenic molecules. Okay, so quickly I'll move on to the key for me is like, well, you know, instead of kind of just going with an assessment of risk based on all these different categories and not really knowing how to bring them together, what might it make sense or how might it make sense to build a framework to bring them together? So quite some years ago, we started an effort to look at building a systems model of the immune system. And essentially we had sort of components such as the dendritic cells. So this annotation here is a bit funny, but it's a naive dendritic cell, a maturation signal, which might be you know, a toll-like receptor agonist or, you know, other adjuvant-like effect to make the mature dendritic cell. And then we've got also through the naive cells, naive T cells, activated T cells, and the memory T cells as well. And then in the B cell compartment are naive B cells and then activated B cells and memory, and then the plasma cells producing the antibody. So we kind of tried to capture the main components. Again, another clear, obvious criticism of this was a bit like the diagram. It seems to be a bit one way from a DC here to a B cell, and we know there's a lot of conversations going on. The whole intent was to try and get something that was simplified that would describe the data we were observing. And we were able to observe from this as well, hopefully no surprise to immunologists generally, but things that seem to be contributing to sensitivity in the assay would be things like the number of T cell epitopes and the binding affinity for those T epitopes. The things like the B cell epitopes and the binding affinity. We tend to, for monoclonal antibodies, really focus on the B epitopes as being essentially the antigen binding site. As I described earlier, it's usually the quite a small area that's actually the foreign sequence, and kind of it sort of fits with some publications around adalimumab and natalizumab, as well as some in-house data we had on a couple of our immunogenic programs that there was really one overlapping area that the ADA would recognize. Within the context of looking at host, MHC class is very important, but things like naive T cell number showed like quite a lot of sensitivity for this assay, for the model, so that if you've got data from an assay here, it gives you a lot more confidence than trying to guess these numbers. A number of other factors related to assay-specific variables, as I mentioned before, and very much driven by regulators, and I know Amy is very passionate about this, getting us to a point where we were actually getting sensitive enough assays that were drug tolerant, so we could be confident that we could detect ADA even in the presence of drug. It is very helpful for us to understand immunogenicity, but also can still mask some of the lower levels. So it's important to have these factors in the model. Just to highlight where we are at present with this, that we see that we've got some assays, population data, we can run simulations, and we've got a consortium. These are the initial partners in the consortium. Certara is sort of central to this as the modeling powerhouse. And we're able to demonstrate improvements in modeling for adalimumab, and indeed picking out differences where we've got, you know, more like the 30% ADA incidence in some European studies as opposed to 70, 80, up to 90% incidence in particularly for some of the biosimilar data. So this is an ongoing effort. Within Pfizer, we looked at a range of our own assets and some I guess biosimilar space assets, and we made some predictions. Largely the ones with the range on from phase 1 studies where we have a range of different doses. And whilst typically the lower dose is lower immunogenicity in the range we were looking at, that wasn't always the case. There's some sort of anomalies in this as well where, you know, we kind of didn't detect ADA, or the model didn't predict ADA at some very low doses for some molecules. Indeed, for Herceptin, it seemed to predict very high, although we've since looked back at the a potential epitope here, and we think that it's probably not real. So, you know, the prediction without the epitope would be much lower. Really key for bococizumab, you know, we were hitting this 50% incidence as predicted by the model, well, observed and predicted by the model. You know, it's not perfectly in line with the kinetics, but when you look at the impact then on the loss of exposure over time, you know, model does a pretty good job of definitely at the population level predicting this. Still some work to be done on understanding some individual outcomes. And again, just to highlight, you know, it can take many weeks, months to really see the impact of ADA in a study. And so what we were hoping is this model would give that insight into an early project team, well, you know, this is a potential outcome later in a clinical trial. And as with any modeling and simulation during clinical development, you expect that you can add the data you find in phase 1 or phase 2 to those then later trials. So, you know, you'd expect that improvements in modeling and simulation, or the simulations, would occur based on that approach. So take-home messages, immunogenicity is a significant barrier to drug development, but the current predictions are actionable. So we can do that both in the molecular design stage and then impacting or looking at the impact in clinical trials. I mean, the simple thing, I guess, like we just heard, you know, remove the T-cell epitopes to avoid immunogenicity is a really good starting place, because we have lots of great tools to do that. And I think I'm going to hear more from Jeremy on that very shortly.
Great, thank you very much, Tim. That's super. We do have a question. I'm going to hand over to Highlim Cho to ask that. Highlim, I'm just going to unmute you so you can ask the question.
Great, thank you, team, for the presentation. So in the case of enzyme replacement therapy, where, like, especially for those genetic diseases, when the patient do not express that endogenous proteins, so do you think we should come up with different strategies for that kind of cases, where, like, those germline sequences no longer tolerable in that patient? So what is your recommendation for such cases?
Yeah, that's right. I think kind of the question of tolerance is an important one, because you know, we make assumptions about tolerance a lot of the time, and I think, you know, even for some protein therapeutics, maybe we're overestimating what tolerance may look like. You know, I think this is really the next big challenge for us, because from a point of view of if we wanted to go into a patient with an enzyme that they don't express in any way, and we've seen that already with things like Pompe's, where there may be no CRIM material. So, you know, it is necessary then to think about, well, what are we going to do to tolerize? And Amy touched on it, but there's a number of publications with tolerizing strategies. None of them are trivial at present, and I think there's a lot more to do to work on what would be a more broadly applicable tolerization strategy. So I don't have the answer as to exactly what that is. There's a lot of different companies working on it, and I would hope we'd start to see some good impacts later or before too long. The interesting question is, do you take the protein that you've got and say, well, it's going to be foreign anyway, so I may as well just do what I can to minimize you know, the T-cell epitopes in that context, and potentially put in some things that look like HLA binders that you do expect tolerance to. And you could end up with very different protein structures than what you're expecting as a starting position. I'm aware there are some companies now who are basically throwing out nature as an architect and saying, well, what's the problem we're trying to address? Let's just make a protein that does that job. And along the way, there's probably a lot of design space where we can design in or out epitopes as required.
Great, thank you.
Thank you.
Tim, for the answer. Daniel asked a question: since the B cell is so critical in the production of anti-drug antibodies, why doesn't anyone assay binding or internalization in B cells as an immunogenicity assessment approach? I guess, why doesn't anybody use assay binding?
Yeah, so it's interesting because I guess it's the establishment of the tools and the robustness of the tools. So we know with most of these DC and T cell based assays that they're pretty well reproducible. You definitely get differences in different donors. We have spent quite a bit of time, and I'll credit Li Shu and Salim Sheikh for working on that over the last few years at Pfizer. And there's some intriguing data that we see. It just doesn't seem to quite be there. One of the area I think that is fascinating, or one system that's fascinating, is the MIMIC system. Maybe one for ProImmune for next year, if you can get someone from Sanofi Vax Design Campus to come and talk about that again, because they basically take the DC T cell assay, and I think because of the way they produce the DCs, it seems that they're long-lived enough to be able to then allow for that downstream stimulation of B cells. I know they're using that more in vaccine studies as a potential replacement to doing clinical trials, but I've not seen for a while anything in the biotherapeutic space from them, but it's really promising.
Great, okay. And then one last question from Seema. Over to you, Seema. No problem, I will ask it for her. On the one hand, immunogenicity is required, which can be related to strong MHC peptide binding affinity. On the other hand, it's not really needed, obviously, to prevent autoimmune reactions. How do we achieve that balance? Big question for you there.
Yeah, I mean, from the point of view of looking at an individual product, then I think kind of everything we do in industry has to come down to what's the product we've got in front of us. So yeah, from my perspective of not wanting the immune response, then we go as stealthy as possible using the kind of tools we've described. And it should be possible that mechanism of action, or you can almost like decouple your mechanism of action. So this is a challenge, right? So if you're generally dampening down the immune system, it may feel good, but actually if you targeting dendritic cells, you could still end up with an immune response. So you definitely need to clean those molecules of T cell epitopes. If you're looking at stimulating the immune system, then perhaps you have to look a bit deeper and draw a tougher stringency with your in silico, maybe load more into your DCs and your MAPS assay than you would normally do. So I don't think we have good cutoffs for that as yet. I mean, in terms of vaccination, then a lot of these tools are built out of the same toolkit essentially. It's just kind of differentiating them, whereas often we want nice strong responses, so there's not for a vaccine, so there's not too many difficulties in using, or at least in getting the signal out of the tools. Usually there, often the problem is actually getting a strong enough or an antibody response that what it does. And again, the lack of B cell tools is something that I think the industry needs to address in the next few years.
Great, thank you very much indeed, Tim. Thank you.
Thanks very much.